Hello everyone, this is my project of 2-week molecular docking and lead discovery internship program. In this assignment, I perform molecular docking of protein EGFR which found associated with brain cancer, glioblastoma and a small molecule gardening to know how they interact with each other by using several databases. Here, we first assess our protein using database UniProt. Here first open UniProt and search for protein by writing or typing its name. Here I take EGFR and there is 3 ET results are found. In next page, we filter out by selecting reviewed and human and we select the first one P0053 protein and click on this. Later on, we scroll down to structure section and there are several PDB IDs. In this, we consider first one 1IVO and download it from RCSB protein data bank. This is the structure of the protein. Now it's time for ligand extraction. For ligand extraction, we use PubChem database in which we search for our ligand gardening. There is a result visible in this. Click on this and there is option of download. Click on download and go to 3D Conformer and download SDF file. Now it's time for ligand and protein preparation. In third step, first we prepare protein molecule using PyMol software. In this diagram, as we see, this is our protein having this red dot containing water molecules and these are extra ligand which is not important for our study, so we need to remove this. After removing, this is our cleaned protein. This is our clean protein. We remove water and ligand by selecting on A and remove water and ligand by selecting each ligand and delete. Now this prepared protein now exported or saved as PDB by going to file and export molecule in PDB file. Now it's time for preparation of ligand molecule. Here we use OpenTable software. We put SDF file of PubChem downloaded ligand in here and convert it to PDB file. Now convert and it's save. In next step, we use both our prepared ligand and protein for docking by using PyRx. Load prepared protein and ligand in PyRx one by one. This is our protein and ligand. First prepare the protein in PyRx. Load the protein PDB file into PyRx by going to file and load molecule. Right click on protein in the display window and select AutoDock and make macromolecule. This convert our protein to PDB-QT format and prepare it for docking. Next, we prepare our ligand. Scroll down and click on Open Bevel and insert our ligand of choice, which is gardening. Right click on, after selecting the ligand, right click on ligand and select minimize all to perform an energy minimization, and after minimization right click again and select convert all to AutoDock ligand. This convert our ligand to PDB-QT format. Now it's time to set a grid box. To set a grid box, we go to AutoDock Wizard, and by stretching these spheres, we cover all our protein to know the docking poses. After this, access the AutoDock by clicking on this Vina wizard and start docking or run dock. After this, after docking completed, a table is generated. This is our table in which binding affinities, mode, RMSD lower bound, RMSD upper bound values are given. In this table, as we see, after completion of docking, minus 7.6 is our highest binding affinity. So after this, click on AutoDock and click on EGFR. These are the different outputs model of ligand which present in this. So I select only output model 1 because this is the best pose and binding affinity. This is our ligand, and save. Right click on this and save this in PDB format. Now after docking, there is a step of visualization of our result. For visualization, first open PDB-QT format protein in Discovery Studio, and later on open output ligand 1. Now copy output ligand 1 and paste in EGFR display. This is our protein and this is our ligand. Now we paste output 1 in EGFR, and this is the pose where ligand bind with the EGFR protein. Now for 3D visualization, we go to the ligand interaction section in left side and show distance and label the amino acid by clicking on this protein and label. These are the different types of interaction showing in this ligand and protein interactions. These are the amino acids participate in this, and this is the 2D diagram. This is 3D and this is 2D. In this interaction, conventional hydrogen bonding, carbon hydrogen bond, pi anion, pi step alkyl bonds are present. And this is the diagram I prepared using PyMol software. Here the same amino acids are shown. Which are visible in Discovery Studio. Okay. After all these studies, we say that EGFR is a crucial oncogene in glioblastoma, drive tumor growth, invasion, survival, and resistance to therapy by activating pathways like MAP kinase, PI3 kinase promoting cell proliferation, angiogenesis, and creating an immunosuppressive tumor microenvironment. On the other hand, gardenin, a type of flavonoids found in certain medical plants, has potential neuroprotective effect and anti-cancerous activity against glioblastoma cell in preclinical study. Above this talking study, it is clearly visible that gardenin and EGFR show significant binding affinity of -7.6. It means gardening has power to suppress the effect of EGFR and glioblastoma. So we use this gardenin in to derive drug molecule to overcome the impact of brain cancer and glioblastoma. Thank you.